Ladies and gentlemen, dear distinguished speakers, dear colleagues, dear friends, welcome to our session today on One Health Avian Influenza Risk Assessment. My name is Gülşah Gabriel. I am head of the Department Viral Zoonosis One Health, and at the same time I'm professor for viral zoonosis at the University of Veterinary Medicine in Hanover. This session will be devoted on discussing a potential pandemic risk posed by avian influenza viruses, which have caused and are still causing large outbreaks in birds and poultry, and are already showing first signs of mammalian adaptation. Before I introduce our experts today, I would like to say a few words on the organization of this session. We will first have the presentations, so all presentations one by one, and at the very end we will have a panel discussion open for questions. So the first speaker will be Professor Dorothy Yeboah Manu. Dorothy is director of the Noguchi Memorial Institute for Medical Research, and she's also professor at the University of Ghana. And Dorothy will speak today about avian influenza in West Africa. Her presentation will be followed by that of Professor Martin Beer. Martin is head of the Institute of Diagnostic Virology at the Friedrich Loeffler Institute in Greifswald here in Germany. And Martin will be talking about avian influenza in Europe and its spread to the Americas. The next presentation will be given by Professor Hiroshi Nishiura. He is a professor at the Kyoto University School for Public Health in Japan, and Hiroshi will be speaking about avian influenza in Asia. His presentation will be followed by that of Professor Ab Oosterhuis. Ab is scientific director of the Research Center for Emerging Infections and Zoonosis at the University of Veterinary Medicine in Hanover, and Ab will be speaking about avian influenza in wildlife. After his presentation, I'll be speaking about avian influenza in humans. And last but certainly not least, Dr. Claudia Garcia Meneses from Boehringer Ingelheim will speak about prevention measures at source. So without further ado, I will hand over to Professor Yeboah Manu to give a presentation on avian influenza in West Africa.
Okay. Good afternoon, and thank you for the organizers for inviting me to speak on some of the problems that the world may face if nothing is done about it. And I will concentrate my presentation on what has been done within the African continent, and will focus more on what my institute is doing within Ghana. So this is the map in terms of history of avian influenza outbreaks in Africa. The first official confirmed cases of avian influenza in Africa occurred in Nigeria in 2006 February. Within 3 months, other countries within the sub-region like Ghana, Burkina Faso, Togo are reported of large cases in birds. And these introduction seems to became endemic for about 2 years, and then there was nothing that was reported again within the sub-region until 2014, when Nigeria again was the first country within the sub-region to report avian influenza outbreak again, and months later they spread to other African countries. And within that outbreak, by 2015, East Africa, for the first time, Uganda and some of the countries reported avian influenza, and it moved further to Southern Africa, and it subsided. But then, since 2020, there have been continuous outbreaks within the sub-region. What are the risks that has so far allowed some of these large outbreaks and maintenance of the outbreaks within our sub-region? One of the key that has been identified from various studies is poor biosecurity measures among farmers and within the population, how we handle poultry. You go to, as you can see, you go to the market. This is taken from a small market in Accra. You see live open display of live birds, and people buy these live birds, send them home, and kill them themselves. There are others also who virtually live with live birds because they have a backyard gardens, and occasionally go to market. As you can see within this last cage, you have different breeds in the same cage from different farmers that they are mixing. So there's a lot of interaction between humans and birds, and also even among birds from the very different farms. And of course, within West Africa, there are cross-border trade. We have people crossing the Togo border, go and buy birds, come to Accra, the Ghana side of the border. And so these are measures that enhance the spread of avian influenza within the sub-region. Have from various epidemics that have been reported within the sub-region, there are evidence to indicate that West Africa and most of the time Nigeria is like a sink of avian influenza carried within the Euro-Asian flyway, whereby birds, wild birds, within winter come to West Africa, mix with local wild birds, and then get transmitted within poultry. And from various genomic studies that have been conducted, a thorough review, you could see that within sometimes one epidemic, there are different introductions, and these introductions are the countries where they are introduced from are in the circles, and they get into West Africa, and then West Africa start dividing and given to other parts of the sub-region. And so until now, various types of avian influenza has been confirmed from different, almost all the sub-regions within the sub-Saharan Africa, from Central Africa, East Africa, Southern Africa, and West Africa. And you could see that in addition to the high pathogenic avian influenza, there are also low pathogenic avian influenza types that have been reported. And if you look at the map on the right side, you could see those in the red are countries where they've at least reported a high pathogenic avian influenza, and some of them have reported both high and low. Those in the orange are those that have the potential, but they don't have the resources, so there has not been confirmed of their pathogenicity. And some also, those in black, so far there hasn't been any such studies that has been conducted. And so moving on into Ghana, our first outbreak was in 2007. You could see from the map that several birds were killed because it's affected more than 100,000 poultry within the country. And this was initially reported in the Greater Accra region, but it spread to other parts of the country within that. And within that space, at that time in country, there wasn't a surveillance system for this, and there wasn't much capacity in terms of laboratory support for surveillance. So following this, the map here between the Veterinary Service Department and the Ghana Health Service as Ministry of Health, as well as the Noguchi Memorial Institute for Medical Research, where I work, came together to form the National Avian Influenza Sentinel Surveillance System. And I might say this surveillance system is upon which we leverage for the national coordination of the COVID-19 response in Ghana. And within this, there were a lot of training, and the training, both civilian and the military and the security were also trained in terms of handling and how to diagnose this. And through that, capacity was built for laboratory infrastructure. So following the 2006-2008, the next outbreak was 2015, and that led to more birds being killed and affected more regions. And that time capacity, and we have a PhD student who was working with GOSA here, so some collaboration between Ghana and Germany was initiated, and were able to characterize the viral strains that caused the outbreak. And we found out that most of the Ghanaian isolates were homologous, and they clustered with Nigeria, confirming that with more than 90% homology, indicating that the isolates were actually imported from Nigeria to Ghana. This started. And one of the things that we found was that within the HA region, we had motifs that could adapt to receptors for mammalian salicylic acid receptors. However, we didn't find the same with the polymerase basic protein 2. Probably this could be the reason why so far with all the interaction that we have between humans and animals, we haven't seen wild outbreak of. But then, in addition to the high pathogenic avian influenza, in 2017-2018, there were farmers who were reporting of death of birds and then also low production of eggs, and this was also taken up, and thus we found out the outbreak was due to H9N2. However, there was surprising with the massive replication of that strain, because this is supposed to be a low pathogenic strain, but with the analysis this was found to be co-infection with infectious bronchitis virus. And so within our setting, this shows that probably even a low pathogenic one could cause more havoc based on co-infection. And this 7 of the strains were characterized, and we found it to be clustering with strains from Asia as well as Middle East. Probably these were introduced into the countries from these other countries. Since 2021, there has been a continuous outbreak, and our institute is on top of it, and it's affected more regions, as you can see. And this has led to, from the Ministry of Agriculture, more birds have died because of this outbreak. And because of that, we have now intensified our surveillance system. There are a lot of sampling going on within the affected areas, and we've been able to sequence some of the strains that is causing these outbreaks. And here again, we find them to be caused by H5N1, and we see some of the clustering within distinct regions, showing there's a high transmission ongoing in our country. Nevertheless, our characterization has shown that none of the viruses has the motives for adaptation to mammalia, which is a bit of good news. The One Health so far, even though we've confirmed from other animals so far, none of all the influenza human cases have all not been to H5N1, and this is good news, and we are happy with it. But this is not something that we should club, considering still the low biosecurity measures that are in place. I would say that based on what is going, avian influenza is of a great economic threat to the sub-region, because now poultry rearing is becoming a good economic measures for the populace, and also a good source of protein. And if you look at the birds, recently I saw a publication in South Africa about 7.5 million birds have been killed. And if you look at what I'm reporting from Ghana, Nigeria has the highest hit within the sub-region, has had several birds that have been killed. This means that this is of cause for concern, an area with a low income, and one of the income measures is suffering. Then there's more to be done. So my take-home message is that yes, there is increased outbreaks since 2006 within sub-Saharan Africa, especially within the West Africa sub-region. Nigeria so far has recorded more than 1,000 outbreaks. Work has been done, and there has been increased surveillance studies, and capacity is being built, especially within the laboratory diagnostics system. However, there's need for increased surveillance among wild birds, because it's only few countries within the sub-region, Senegal, who have been able to do. Even Ghana, we are not doing much in terms of wild animal bird surveillance, and we need to do more. And we need to increase also our One Health approach for surveillance, because of the interaction between the environment and the human population. We need to do more. And of course, there's opportunity. We see this capacity as opportunity to use for other diseases, as was used in Ghana during the COVID outbreak. This our work from Ghana. The champions are Professor William Ampofu and Dr. Ivy Asante, and I would like to acknowledge them because our institute is one of the WHO regional laboratories, and these are the ones who are heading them, contributing to data to the global the Gazette. And this work within our institute is a collaboration between the health service and the veterinary service, and I would like to acknowledge everybody who has been part of our institute. And thank you very much for your attention.
Yeah, thank you very much for the invitation to present here the situation in Europe. So I will concentrate only on the highly pathogenic H5 virus, because I think that's enough for 12 minutes. And, and we had enough to do. And I'm working here on the island of Reims, that's the Federal Research Institute for Animal Health, and we are also the VOA and FAO reference lab for avian influenza viruses. So that is something you heard in the last years nearly all the time, worst ever bird flu crisis in Europe. And this is a good summary of the situation we had since 2016. There was an ongoing high-pass influenza virus outbreak season. We had one clade which took over, and I will show you this in more detail, the clade 2344B, so that's the code of this virus. It's reassorting, so this is a virus which can mix with other influenza viruses, and it learned to do this as good as possible. And we have a wild bird outbreak situation which is connected to poultry and mammalian spillovers. And since 2021, and this is one of the take-home messages, this disease, and we never believed that before, is endemic in Europe. So the virus learned to have the situation here in Europe, like in other countries, like in Asia, where we knew this, and it can survive here also over summer. How did this start? And you might see this in later presentations also. It's all going back to the virus which is called A. goose Guangdong 1996. So this is a 27-year-old story. And the virus took this time to improve further and to get what it could do, first in 2005, then again in 2008, 14, 16, and 21, coming from South Asia to Europe. From Europe and also from South Asia going to Africa, we heard this. It's more or less the same viruses we share here. And then this virus was improved further, and in 2021, it took this transcontinental spread going to Northern America. And then you have a completely new field for this virus to spread further. This was seen before from the other side in 2014 for a similar virus, but then it stopped. It did not go to South America. But this time it made it. So the virus further improved. It could jump to South America, and this is the first time this kind of virus is ever reaching this continent. So a completely new animal world getting in contact with such a virus. So it's really it's kind of an experiment. We look at what the virus is doing there, and the consequences on one hand were we learned that also for the first time Iceland was affected, and it was one of the stepping stones to get the virus from Europe to North America. And overall, we have severe impact on both poultry and biodiversity. So there is protected species hit, there is colony breeding, birds, tens of thousands dying from the disease. And if you look only at poultry, it's now far more than 150 million poultry dead or culled, only in Northern America and in Europe. So this is numbers we never had before. So this virus 2344B, that is the one which took over in 2016, is the one which could do this. But there is white spots on the map. So one is Galapagos Islands, which is hit now. So the virus made it also there. And the next step is, unfortunately, probably Antarctica, to go there. And other islands in the Indian Ocean, for example. And still, it's not in Oceania. It's not in Australia. So we will see. But the virus has some power to spread. We saw this, so it's probably a matter of time. So a situation we never had before. And this started, as I said, and we can look here for this genetic analysis. This is the grandfather, the goose Guangdong virus. And then this HA, the hemagglutinin, this gives the clades names. It goes down here, and you have a lot of different clades. You have seen this also in Africa. And this 2344B is the one. And here you see, you know this from SARS-CoV-2, what kind of virus is taking over. And you see from 2016 on, this is a major and dominant virus in many parts of the world, and especially also in Europe. So it started with one of the clades in 2005, 2006, which were not able to reassort a lot. So this was surprising that this changed then in 2016. And you see all these viruses, H5N8 was the one which came up. And then you have an N5, N1, N3, N4. So the virus was, it's a kind of a training. It was mixing with all the viruses which are in wild birds, the low-pass viruses. And then it decided again to stay in an H5N1. But this is still reassorting. This is only one name for a lot of viruses. This is a cloud of viruses. And this is the outcome then there, the wild bird cases from 2016 on, and you see there is even no space on the map anymore. So there's so many wild birds infected over the time, but you can nicely see here, it started with H5N8, and you always have a gap. Then it moved to H5N1, and you don't see a gap anymore. So we will go into detail there again. And if you have a lot of wild birds infected, this is prey for carnivores. So you get much more interface between birds, infected birds, and other wildlife animals, especially mammals. And the highest risk we see there is the fur farms. So for the first time, we see really large outbreaks like in Finland with dozens of farms where the virus is spreading. And it is the virus coming from the wild birds. And we will hear later by Ab Osterhaus what is the mutations we follow there. The number of affected wild birds is as high as never before. So nearly every kind of bird, but especially its ducks, geese, and gulls can be infected, and it's must-die-offs. Here you have barnacle geese and the British Islands. You had a lot of geese and ducks in the Wadden Sea in Germany. And we are talking about tens and tens of thousands of birds, which are migratory birds, resting areas. And then you have birds of prey eating there or feeding their young stock there. And this is close to our island. This is 2 young wild-tailed sea eagles. So there is more and more birds of prey, which we use as sentinels, because they show you first where the virus is. And we have more than 100 affected wild bird species up to now. It's getting more and more. We even don't know in South America what kind of birds are now really infected. And what has changed? So this is really new, and this is the enzootic cycle. So the virus stays over the summer. And as I told you, this was the H5N8. And in summer and over some years, it has only to be reintroduced again from South or East Asia. And then we had an outbreak here again. And in summer, it went down. But a little bit was still there. So we were predicting probably the virus has learned to over summer, we call it. And then the next year and the next year, it's even getting worse and worse. So the virus stays here, and it goes into colony breeding birds. You have always connected the poultry outbreaks directly. When you have a lot of wild birds infected, you have also poultry outbreaks, because you cannot completely close a stable, or backyard farms always have contact. And we have a switch in the genotypes. This is the reassortance. And in the last 2 years, one type, it's called the BB type, took over, and it's in Europe. It's a very interesting virus. If we look at it closer, this is a different wild bird species. You only have to concentrate on the green ones here. This is all gulls. What had happened? In 2022, the virus mixed with a gull-type influenza virus, an H13, a very special virus. It took 3 of the 8 segments, and then it switched to a completely new reservoir host, the gulls. And then we had all these outbreaks in colony breeding birds on the coastline. So there you could follow that the virus really learned to go into a new host and spread very fast in this host. And then you had all these terns here, sandwich terns in the Wadden Sea. So tens of thousands of terns die. Gannets, You all know the bus rock, for example, where all these animals live, and a lot of them died. And again, we have this feeding. This is an great owl nestling, which was tested positive, and you can guess where he got the virus. So that was the feeding with all these colony breeding birds, and they breed in summer, and therefore you have all these outbreaks. So a situation which changed now from year to year with different variants, and a virus which is spreading like never before. And this is from the EU reference lab, for example. So we had the AB type in the beginning. This was more in ducks. And then we had this BB type. This is the gull virus we have now in Europe. But you see there's many others. Sometimes it's only one detection because they mix and mix with the viruses we have in the wild birds in the low pass. So we have now more than 40 genotypes. So if we talk about H5N1, it's not one virus. This is dozens of genotypes, and all of them have their own features. So it's very difficult to predict this. And I'm not talking about the North and South America reassortants. There you have the same, reassorting with the viruses which are there. So we have now a big cloud of H5 viruses, and we have only a few characterized up to now. And all this is connected to poultry cases. So a lot of wild bird cases means poultry cases. And you have hotspots. And this is where the high density is in Europe. This is, for example, in France, the duck production, in Germany, the turkey production, in Hungary, the geese production. And this means 50 million and more dead and culled poultry in the last 3 years. So this is really a lot, and it's different in the different countries. And again, it's H5N1 now, and it's exactly the viruses we see in the wild bird. So summarizing this about the H5, it's clade 2344b, and it's a panzootic. So a pandemic in animals. So it probably will take also over in other countries. It's perfectly wild bird adapted. So it's a virus which learned to be better and better in wild birds, but this also leads to spillover, poultry, mammals. It has a severe impact, I showed you, and this is then the secondary spread. If you have it in the system, you might not stop it so easy. And you have all this reassortant, so the virus might change very fast. New is it's endemic now. We have this must die off in colony breeding birds, and we have introduction into firm farm. So we have new features we have to have a look. And if I have to say about the risk, what is an increased risk? Endemic situation, genetic plasticity, continuous spread, and mammalian spillover. So we get more and more features which make a higher and higher risk. Still, the virus is a bird virus. It's perfectly adapted to birds, but this pure number of infection gives the virus, due to the genetic plasticity, some chances. So we have to be aware. Sounds easy, but it's not so easy. Prepared. Prevention measures, biosecurity is one of the most important in all these farm settings. And we will hear later on what can we do. What is with vaccination, for example. With that, I thank you very much. I thank the whole team, a lot of collaborators, and especially also the funding, like the very new project we have here in Europe, EU-funded Kappa flu, where we want to look in closer detail. Thank you very much.
Okay, following Martin, I'd like to briefly overview what's going on in Asia. Yeah, it's coming. Yeah. So, if you look at this figure, you understand why I'm here to represent Asia today. This is a map showing the cumulative number of confirmed human cases with highly pathogenic avian influenza, especially H5, except for Egypt in Middle East. The majority of human cases have been seen in Asia, especially in Southeast Asian countries, most notably Indonesia, Vietnam, Cambodia, and Thailand, and more recently in China. Yeah. And this figure is the cumulative count up to 2016. And if we update that figure from 2017 onwards, the situation didn't change much. Yeah. This figure doesn't include 2, 3, 4, 4B. Yeah. And as you see, that increment has been seen mostly in Asian countries, and other countries with some human cases were mostly seen in the countries with previously known outbreaks in poultry. And one of the reasons which is very evident to us is that Southeast Asia is closely, the intensity of the epidemic is perhaps a little bit higher in the bird species, and also the lifestyle in Southeast Asia is very, very in a close proximity to the chicken and the ducks for eating. For instance, even sometimes within the household we see the chicken. And therefore, the wildlife ecologists have frequently focused on the differential flyway of the wild birds to describe the different types of the evolution of the H5 viruses. And in Asia, East Asian and Australasian flyway has been seen, and only in Arctic region was the area where the wild birds may interact from each other. And as the genomic studies demonstrated, there has been the very limited share of the genome between the European, African migratory flyway and the East Asian and Australasian flyway. And so the epidemiology has been classically very different, but something strange is taking place now. And before moving on to that issue, there has been a very good study taking place on mapping of the risk of the human cases and also the poultry outbreaks to be seen in Southeast Asia. So not only the lifestyle-related risks among humans, the common agroecological conditions have been known as the risk that explains why the Thailand, Vietnam, Cambodia, and other surrounding countries frequently experienced the outbreaks. So ducks are highly abundant, and outbreaks were always seen where the human population density is extremely high, and importantly, rice cropping intensity is high. Is one of the important conditions where we frequently see the infection in the duck species, and not necessarily the outbreak locations was associated with the number of chickens. That's what we have understood classically, and interestingly, that classical issue has been somewhat over prior to the COVID-19 pandemic. The left-hand side figure shows human cases up to 2019. Yeah, and the 2015 spike is what it was seen in Egypt, but except for that, the decreasing trend of H5 influenza in humans was evident, while the transmissibility was more alarming for the humans. That's what the right-hand side figure shows. That more densely colored part is where the clustered infections were seen in humans, while the light or gray colored bars represent the non-clustered infections. And compared with the H7 influenza, H5 influenza was more frequently seen within households, and therefore the household-related transmissibility was suspected to be a little bit higher than that of H7 influenza. And why the epidemic level has been waned is perhaps explained by a single human factor, namely the vaccination. So this figure shows how China implemented vaccination as a function of time. And not only China, but Indonesia and Vietnam are known to have carried out a continuous vaccination practice, mostly focusing on the poultry, and also sometimes ducks for food were also vaccinated. And exact causal association between this vaccination and the decline of the epidemic level of the influenza has not been demonstrated yet. That's how I understand the ongoing problem. But as you see that the doses, total doses of vaccination was in China, it was about always 17 to 18 billion per year. So tremendous number of vaccinations with rapid cycles was what it was required, and therefore the economic benefit to implement that vaccination was once questioned, but that's also not perfectly answered. At least the epidemiologically, the seroconversion among vaccinated chicken have been sufficient. That's what it was demonstrated. But the relevance between the decreasing epidemic level and vaccination has yet to be answered explicitly. And as Martin explained, the things have drastically changed following that decreasing trend due to the appearance of the 2.3.4.4b. And if you look at the HPAI cases in animals from 2022 to 2023, as you see that Asia is already, what it is not our concern. And as you see the epidemic curve, this is from ECDC. The recent development with the emergence of what I should say that it has a resurgence of the H5 due to the reassortment is mostly attributed to 2.3.4.4b in Europe. And looking at the recent molecular study from the University of Hong Kong, what the phylogeographic study has shown is that from Asia, the movement of the virus was seen to the westward to Europe from 2014 to 2017, and that movement of the evolution continuously was seen in H5N8 from 2017 to 20, again, from around the Middle East to the European region. And once the H5 emerged, that became more widespread, and it went to the North America. Now that among the researchers, the epicenter of the HPAI H5 is regarded as being, beyond the Asia, and as it was expressed by Martin, it may be called the panzootic, and Asia may look like not the focus any longer, but now we are seeing the nightmare in Japan. Yeah. From 2020 to 21, the H5N8 outbreaks started in the poultry population. That was historically the largest season, but in 2020 to 23 season, it replaced the record, the historically largest outbreak caused by H5N1, and this is all due to the 2.3.4.4b is seen, and the blue stars and what it was seen in wild birds, while the red circles were in chicken. And the first date we see the outbreak is now much earlier than the usual seasons, and number of outbreaks and the number of slaughtered is, we are already starting to see the H5N1 epidemic in this year, and it's predicted to exceed. And in the Far East Asia, we haven't seen the infection in the wider range of the host, for instance, aquatic mammals, but infection in red fox and so other species on the land are also seen. Not only Japan, but South Korea is also now struggling with a massive number of outbreaks. As you see here, it's even difficult to count, but H5N1 outbreaks is historically largest, and the geographic patterns of the epidemic is exactly the same as what we see in Japan. And as a response, the Japanese government decided to manufacture pre-pandemic vaccine for the first-line workers. Classically, we have started to manufacture that using the Anhui Credo 2.3 virus for the 10 million people. And that was followed by some update of some Credo for 5 million, 3 million, 9 million, and so on. But they are all expired now. Yeah. And we started to prepare against the H7N9 from around 2020. But this year, looking at the phylogenetic evolution of the 2344B, we have had to update our notion, and we decided to prepare using the Astracan 2344B virus to manufacture vaccine. And it's going to cover at least 5% of the entire population, namely the target is the first-line workers and the self-defense force members and those working for the emergency service, maybe. So summarizing what I have briefly talked, so decreased incidence of a classical picture of HPAI, H5, has been seen in Asia. Yeah. And the exact reason for the decline remains unclear. And what we have to remind is that whether that vaccination practice gave some selection pressure for the further evolution is also what to be studied closely in the future, I guess. And recently studies suggested that the epicenter of 2344B has been expanded westward, but it's now coming back to Asia. And no evident aquatic mammal outbreaks yet, but historically largest outbreaks are going on. And pre-pandemic vaccines are, acting as a kind of the wake-up call. That's all from me, and I'd like to pass the microphone to Professor Albert Osterhaus. Thank you very much.
Well, first of all, thank you very much for the invitation. I'm going to talk mainly about influenza in terrestrial and aquatic wildlife. And you've heard from Martin already that a lot is going on and that we're really suffering from a real pandemic in animals. And the question is, can that spill over to humans? That's the big question at the moment. Well, what you see here on this slide is basically how this whole thing is happening. You see here, in the center here, you have the migratory birds. And we have seen that originally it came from Asia, and then spilled over to the birds in humans, to the domestic birds, and then we got the big outbreaks there. But now, as we have heard, we have really got an endemic situation. Well, this is a very old one, and Martin already alluded to the Guangdong situation there. When we first identified the first human case of H5N1 in Hong Kong, nobody believed us, because they all said this is not possible. You have to go back to your textbooks, because these viruses, they do not spill over to humans. And as a matter of fact, we learned our lesson. And in that case, after the first case, we had 17 subsequent cases, 6 people died. And as a matter of fact, we were all afraid that was going to be the next pandemic. And we are in a rather similar situation at the moment. We've just heard that already we were making pre-pandemic vaccines. We are doing, in close collaboration with industry, we already were doing phase 1, 2, 3 trials, and really to prepare for the pandemic. And perhaps a lesson there is you can never predict what's going to happen in flu, because we got the H1N1 from Mexico. So completely different story. Well, just from history, you see here a paper we have about the H7N7 outbreak in the Netherlands, which really was an outbreak in poultry that spilled over repeatedly to humans. As you can see here, we had 1 fatal case at that time in the Netherlands. So basically, and we did a lot of work on that. The crucial thing there was all the time that this virus did not spill over from human to human. So this was just a one-off infection all the time. And we did a lot of work to show what mutations, especially the H5N1 virus, would need to become a transmissible virus. But I'm not going to talk about that. Here, the H7N9, you've just heard that. Yeah, so that was stopped more or less by all the measures that we're taking, including vaccination and a number of other measures at that time. And that basically it's still simmering on, but it's still there. But basically it's not the biggest threat. We are talking about the H5N1 at the moment predominantly. And then we're switching gears again, because I'm talking about wild carnivores basically. And you see here another example, a study that we did in close collaboration with our previous lab in Rotterdam, Ron Fouché's lab. And as a matter of fact, what we showed is that we had an outbreak that killed about 20% of the seals in northwestern Europe, so up to 20%, and that virus spilled from seal to seal. And we were able to actually follow it from here to there when it actually disappeared. The virus disappeared. We don't know why, but as a matter of fact, we did a lot of work on sequencing those viruses. I'm not going to show you all the details there, but it was really we clearly showed that from a really avian virus, it adapted to the mammalian species, really by a whole series of mutations in the receptor binding domain, basically in the hemagglutinin. So basically, it's not what we are talking about today, but it really shows that you have to be prepared that these things can happen also in terrestrial, but also in aquatic mammals. Well, we have seen a lot of this, and the migratory routes. As I said before, we started with a virus that came from Asia and moved over every winter time to the Netherlands, Germany, and our West European countries. And that happened every year again. But now we have really got an endemic situation, which is completely different. One of the studies we did is perhaps interesting to tell you, is that where we saw the outbreaks, we could actually predict that, because there, and that was at that time, there where the water was not frozen, so you could just follow that. There we got all the big outbreaks, because the migratory birds would go down there and then they spilled it over. It was spilled over to the poultry population. But basically the situation is quite different today. We are much more in a pandemic situation, as we have seen. And this is pictures that were shown by Martin as well. And this is the birds, both the wild birds, where you have all these different species. And it's this H5N1 going on and on and on all the time. And the question is this going to stop? It's endemic in many countries. It's really a virtual soil epidemic at the moment, which is really, if we say it's a pandemic in animals, we'd be a little bit careful, because as you saw from one of the slides of Martin, he had an arrow going into Australia. As far as I know, it has not been demonstrated in Australia yet. So formally, we cannot talk about the panzootic yet. But I think we're very close to it, and it's probably going to happen. We have quite a bit of experience with H5 and 8, so the earlier virus that you saw, spilling over to seals. And we got, in this case, on your left-hand side, you see here 2 seals here in Germany that we picked up. And as a matter of fact, we identified them really as H5 and an avian virus. It's completely avian. The interesting thing here, these animals did not or barely developed a pneumonia. The virus went straight into the brain. And we don't know. They died from an encephalitis. One of the animals, of these 2 animals, had also a starting pneumonia, but it died before it got a real pneumonia. So quite an interesting finding, I think, that you have encephalitis there all the time. So in this case, 2 cases here. And that's the H5N8, as you have seen before in 2020. So more recently, we just published two other cases you see here from the Netherlands and Germany. And this was all worked together with the FLI, with Martin's group. And as a matter of fact, we picked up 2 seals that died from an encephalitis, again, without a pronounced pneumonia. And the interesting thing is that these were individual cases. So we are really looking very carefully. We have a monitoring system for seals in the Netherlands and to some extent in Germany as well. And this is what you see here is the serology. And the top one here, you see the serology after the H10N7. You saw there were antibodies there, both in the harbor seals and the gray seals. So quite a percentage of the animals had this, and the virus was actually spreading at that time. So we were really wondering if this was going to spill over to terrestrial animals, etc. Did not happen, fortunately. The last 2 seals I showed you from Germany and the Netherlands, what you see here, we did quite an extensive serological screening. So all the animals that are being brought into the seal sanctuaries, they're being sampled, blood sampled, and we test them regularly for different viruses, including the H5N1. And fortunately, they're all negative. So the virus is not spreading. Similarly, we tested some 200 or 100 seals here, the brains of these animals and the lungs, of all the seals that are being brought in the northwestern area there, that are being brought in dead. And as a matter of fact, we checked them, they're all negative as well. So in seals, it's not spreading at the moment. That's good news, but you've seen this one before, and you've seen this one before, and what you see here is all these terrestrial animals now, and the aquatic mammals as well. And what you see here is quite a number, and it's really going on, and the question is if this is not just the tip of the iceberg, of course, but because we are just looking into those animals. There's a large monitoring system ongoing in the Netherlands, in Germany they do a lot of work, and then we see again in aquatic mammals, there are indications indeed that we have animal-to-animal spread in the meantime, a little bit similar to what we saw with the H10N7. So this is really something to be worried about, because this is adaptation to a mammalian species, so be careful there. And there are, of course, contacts with humans there. And you see the question mark here on the slide. Is this going to spill over to humans? You can always say there's little contacts between humans and these aquatic mammals, or humans and these terrestrial carnivores like foxes and what have you. And I think on my next slide you see a survey of what we have to date. And it goes all the way. You see the jackals, you see all these different terrestrial species. We see, of course, some of the fur animals here that Martin was referring to, but also when we look at the aquatic mammals, look at that. There's quite a number there in the meantime. And I'm not even talking about the carnivorous birds, the raptors, that we saw before as well. So basically, there's ample opportunity for adaptation to the mammalian species. I think that's the word of caution I would like to say. So there's nothing new in influenza in seals and dolphins and whales. There have been outbreaks all the time. And the question is indeed, because I'm talking about these aquatic mammals, what the heck, who's caring about those? Because they're the terrestrials as well. But a lot is happening there. And then this is a slide I got from Martin, just as an overview here. You see it, and I'm summarizing here the different species where we see it. And we see here to avian influenza here to domestic cats in Poland. You see here another one in South Korea through pet food. And we see H5N1 in Finland, in fur farms. So this is happening all over the place, and it gives ample opportunity to the virus to adapt to the mammalian species. A similar story that we have heard before about COVID-19, so where we had a similar thing, and they had to cull all the fur animals. And of course, you have to look for mutations there, and that is the difficult thing. And of course, you see here also these are captive animals, so fur animals. And this is a picture I got from Martin. So really there are contacts with people do swim with dolphins and with sea lions, etc. So this might not be as harmful as it all. Well, I'm more afraid of these animals in the first place, but anyway. So there are other reasons not to do that. So here really what we see is, fortunately, we do not have a human outbreak of this clade of H5N1. But we do see the occasional adaptation, especially in the PB2. It's not the same thing we have seen with the H10N7, where we got a real seal outbreak. There you got mutations in the hemagglutinin as well. We know in the meantime from the work we have done in the past on ferrets, where we artificially did really had the virus transmitted from ferret to ferret and looking at the mutations. So we are very keen to do that. But you see at least some of the mutations popping up. You see here the 627K mutation here and the 710N mutation here, and that is in the wildlife. And these are domestic animals. So the flu is really knocking on the door. I think the H5N1, we should agree that it's knocking on the door. And basically, we have seen this before, and this is basically a very good news. So this clade of virus is still not adapted to humans. So that's the good news. We don't see that yet. But of course, we have to be very careful there. But you see already we are preparing for a next pandemics or pre-pandemic vaccines. And as I told you before, we did the same thing just before the swine flu that originated from Mexico. And then it became an H1N1. So I'm not going to predict it's the H5N1, but it is a real possibility. So I think really preparation for the next pandemic, being able to do that is absolutely important. Well, and this is from ECDC. I think it's important to see that they have advocated for an increased surveillance of people in hospital who do come to a hospital with either a pneumonia or an encephalitis that cannot be explained. To really test them for avian influenza, or at least for influenza, because this is not the classical, especially with the encephalitis. If you have an ununderstood encephalitis, go for the anamnesis and really make sure that it's not influenza, because adaptation to humans in hospitals is the thing that we are worried about. Okay, this is what I wanted to say. I would like to thank my own group, but then the SEAL centers that have been working very hard on this, and of course Martin Beer and Tim Harder's group. Thank you very much.
So I would like to focus on H7N9 influenza, which Ab has briefly touched on. H7N9 influenza is just another example of an avian influenza virus that has cross-species barriers and infected humans, not only having infected humans, but has caused large outbreaks, several outbreaks in humans. H7N9 influenza was first detected in humans presenting lower respiratory tract infections, leading to pneumonia and eventually death. The causative agent of these lower respiratory tract infections were identified to be these newly emerged H7N9 influenza A viruses in East China in 2013. And this virus harbored gene segments of various bird species and already showing some mammalian adaptive signatures throughout the genome, which suggests an increased risk of avian mammalian adaptation. And it became also soon clear that the major risk factor to get infected with avian influenza is having close contact to infected poultry. And this hypothesis was further strengthened by the fact that closure of poultry markets in China was associated with reduced human cases. So there were 5 subsequent waves with high incidences in humans, leading to lethalities between 30 and 40%. And the last incidence in 2017, also the virus had turned from a low pathogenic avian influenza virus to a high pathogenic avian influenza virus, initiating a massive vaccination campaign in poultry in China, which proved to be very successful, strongly reducing human infections. If we have a closer look to these 5 subsequent waves observed in humans who was at risk, it became evident that the incidence is highest in males infected with H7N9 compared to females. There are several factors which are associated with sex differences in disease outcome. It could be behavioral aspects, such as males are more likely to visit poultry markets and thus being exposed to infected poultry and getting infected. Or there might be yet unknown host factors that affect disease development differentially in men and women. And another hypothesis is that sex hormones could drive or contribute to disease development in males and females. And sex hormones can, of course, vary depending on age, and also they can vary depending on comorbidities. So we hypothesized that we were interested that we would like to study the role of sex hormones, which might play a role or contribute to this severe disease outcome observed in H7N9 influenza in humans. And I would like to highlight that sex hormones are not only required for reproduction, sex hormones are actually inducers of so-called sex hormone receptors, which are nuclear receptors acting as transcription factors and thus regulating the expression of various genes from head to toe, including those involved in antiviral immunity. So we teamed up together with Yualong Xu from the China CDC in Beijing in 2017. And there was my team coming together in Beijing together with Yualong Xu's team. And we received access to a large avian H7N9 cohort, which we have jointly then studied regarding the role of sex hormones in H7N9 disease development. And this cohort was, of course, controlled by H7N9 negative cases, by close contacts to those infected with H7N9, as well as poultry workers, but also we had control cohorts of patients infected with seasonal H1N1 or H3N2 influenza, either mild or severe cases. And to make a long story short, we have measured several sex hormones and their regulators, and we ended up with observing that particularly low testosterone levels in H7N9 infected men were associated as compared to H7N9 negative cases with a severe disease development. And during the course of infection and hospitalization, low testosterone levels were associated with an increased risk to die of H7N9 influenza. Sex hormone levels were not significantly altered in females with H7N9 influenza. We also detected for a various panel of cytokines and chemokines and found, similar to what others have shown in this cohort, that an increased expression of inflammatory cytokines and chemokines is associated with severe disease outcome in males. And I would also like to mention that some of these cytokines and chemokines have in their promoter region binding sites for sex hormone receptors and might be either positively or negatively affected in terms of their transcription. And we found that two cytokines are associated with low testosterone levels and death in H7N9 infected men. IL-6 is one example, and particularly IL-15 showed the strongest correlation between low testosterone levels and death in H7N9 infected males. So in the human cohort, this association did not allow us to make any potential causal relationships between the infection itself and the testosterone levels. So we used a preclinical animal model. We've infected male mice with H7N9 influenza, and we could observe that the infection itself in the infected male animals leads to a reduction of testosterone levels in every so the post-infection levels, circulating testosterone levels were lower compared to pre-infection levels, and that was observed in every animal, in some of them moderate, in others quite strongly, but reproducibly in every infected animal. Post-infection testosterone levels were far lower compared to pre-infection testosterone levels. We could also detect that the virus infects and replicates in the testes of the male animals without affecting gross pathology, suggesting that, leading to the hypothesis at least, that infection of the testes and replication of the testes, so the major organ producing testosterone, might have an influence in reducing testosterone levels, which could then have an overall effect on overall health, actually. So this is a newly emerging research area that infections can have an endocrine hit, affect the HPG axis, and one might now speculate whether this could lead to any long-term consequences in these infected patients. And so while we were compiling data and studying and discussing with our Chinese partners, we found ourselves in the beginning of the COVID-19 pandemic again having a déjà vu that males are more likely to undergo severe disease compared to females. So we immediately translated this finding in close collaboration with partners at the University Hospital Hamburg-Eppendorf. We have analyzed sex hormone levels in patients who required intensive care. And similar to H7N9 influenza, we observed that these patients have low testosterone levels, but in contrast to avian H7N9 influenza, this was paralleled by increased estradiol levels. And very recently, we have identified a gene called CYP19A1, encoding for the enzyme aromatase, which converts testosterone to estradiol. We identified a mutation herein together, jointly in collaboration with the University of Siena in Italy, finding this mutation particularly prominent in males hospitalized with with COVID-19, and particularly so 70%, almost 70% of all males hospitalized with COVID-19 did show this mutation. And there were also other brilliant studies from colleagues from Milan University in Italy showing that COVID-19 patients, even after recovery, show low testosterone levels. They did a wonderful follow-up study showing that 7 months after in recovery, still 50% of all males were hypogonadal, so showing low testosterone levels. And even 1 year later, 30% of all males were hypogonadal, now leading to the speculation whether this could have any long-term consequences regarding overall health. And with that, I would like to thank all of our partners throughout the globe, and would like to highlight that global collaboration is absolutely essential to understand disease, so how infections cause diseases, which might pave then the way in designing and discussing potential therapies. Thank you very much.
Now is my turn. Sorry, I had mixed it up. Good afternoon. I work in the poultry industry, and I can tell you your poultry meat or eggs are safe. And I actually made some chicken at lunch today. But I'm serious. The effort that the companies producing meat and eggs are doing to control and prevent this disease is enormous. And the support they're getting from their government officials is also paired to none that we have seen. They're taking this very seriously, as well as we do. We heard loud and clear that avian influenza is a very difficult situation for us in the poultry industry, because this not only has implications in the food security and the availability of food, but also it does have an emotional well-being for farmers, because they need to see how their birds are culled or depopulated, but also they have to fill out a tremendous amount of paperwork in some cases to receive compensation for that exercise or not. And they know that they might not be able to repopulate their farms on time. And also we heard today that could lead potentially to human pandemic. As I told you, governments and policymakers are rethinking how to approach this disease, how to control it, and the most important thing is that they are considering a multitude of things to address it. One of those includes increased biosecurity in the farms, to be sure that the chain remains completely safe. We have seen a lot of maps today, so this only represents for you something important in poultry. There are 3 diseases, different diseases that matter. H9N2, H5, H1 as we heard, and H7. It's very complex how these viruses move and infect to other species. For poultry, these are the 3 diseases that matter, and for those diseases, there are vaccines available to use in poultry farms. When we think about vaccination, vaccines can do these three things. They can increase resistance to the virus infection. They can reduce the replication of this virus in the respiratory tract or in the intestinal tract of the birds. And they can prevent the illness and the death in those populations. So that's why vaccines are used. And when we do that, we can reduce environmental contamination. We heard today that you can get contaminated by direct contact with direct, like touching someone or a bird that is contaminated, or indirect contact by touching something that was contaminated, like a shoe or something that was in contact with a contaminated surface. It can then reduce the spread between premises and the transmissions to birds. Very important in areas where there's not production of birds. In completely closed farms, it reduces the livelihood, it maintains the livelihood and reduces the issues with food security in areas where the farmers are open or that are in the range, they're outside, and it reduces that risk. It also improves animal welfare. Nobody wants an animal or a bird that was sick in the food chain. And that's our job, to secure that the animals that come to the food chain are safe to eat. The limitations, of course, of vaccination are many. I'm just summarizing here the ones that are the most important. When you start vaccination, you might actually have an impact in trade, especially with the H5 and H7 variants. We do know that the World Organization of Animal Health has guidelines very clear that stipulate how to behave or how to treat populations that have been vaccinated. Still, countries have the right to decide how to manage their trade. It also requires a lot of surveillance, and this can be complicated and expensive for some of the farmers, but also the governments that are putting this in place. And it makes the diagnosis more complex if you don't use a vaccine that allows you to quickly differentiate from a vaccine virus or subunit from an actual wild virus. When we look at the vaccination of poultry as of October of this year, you can see in the map in dark blue where the vaccination for H9 is going, and we heard some examples today. Also where the vaccination for H9 and H5 is going, and the combination of H5 and H7 vaccination, different types of vaccines. You can also see where there's no vaccination allowed for avian influenza at the moment. We heard today some cases about human infection, and you can see in the graph on the right, the human infections correlated with the years, and the size of the bubble represents the cumulative numbers of cases. So you can see what are the types of viruses that are causing more problems or could cause more problems with humans. We know that the avian influenza viruses do have the capacity to transmit the disease and to cause a zoonotic problem. And we have seen in specific countries cases where vaccination has reduced this zoonotic problem. Two examples that I'm going to show you. The first one is the vaccination against H9N2. In this case, you can see what you heard today. There are two different types of viruses in China that come from the same lineage. And you can see that when vaccination is secure, and they have different types of vaccines in China, there is a reduction in the number of cases. H7N9 vaccination in China as well. Once we started seeing an increase in vaccination of birds, there was a clear reduction of cases induced in humans. We do not have, that I know, data that proves that when you vaccinate with H5N1 or N2, there's a clear reduction of cases, but there's an example of Mexico, which has vaccinated with H5N2, and then they were able to reduce the wild virus or the virus completely. As I mentioned before, there are different types of vaccines. I'm not going to go through all the vaccines that are available, but I can tell you that a meta-analysis that was done with the different vaccines that are available, those can be inactivated vaccines that are injected in birds, or they are recombinant vaccines with H5 inserts, or it can be non-replicating mRNA vaccines. All of those can reduce the replication of the virus, and they can also reduce the mortality in the birds. So, in some cases, some vaccines have the opportunity to help you differentiate which one is the virus from the vaccine or from the field, and this represents that example. This is called a DIVA approach, and you can see how you have an infection and you have antibodies produced by the birds against those. Once you vaccinate with a vector or subunit vaccine, in this case, the bird will create antibodies against this hemagglutinin. Once you get an infection, then you will have, or there's another viral situation going, you can see how there's a very quick and strong development response for this antigen, and this is how you can very quickly control and know what type of virus you have, but also to see the reduction in signs on mortality of birds. As I mentioned before, there are multiple types of vaccines, but a vaccine is not enough. A vaccination program always requires a 360-degree approach to biosecurity. And I can tell you that the key companies that produce the eggs, the meat that we consume around the globe, have a very serious biosecurity response. They have increased tremendously how they do this. And what remains very critical is the free-range populations that are difficult to control, right? That they're outside, they can be in contact, direct contact with wild animals. And those are usually what we call the sentinel birds, because they get sick and we know they're close from where the commercial farms are. I'm going to close this presentation by letting you know that our company, Boehringer Ingelheim, is a company that is leading in pharmaceutical products, in vaccines as well. We are very seriously working to support the global effort against avian influenza. We're committed with not only producing vaccines that we have and are used now commercially, but also to continue to bring new innovation that adapts to the needs of the virus that we have seen. And this is a science-driven company, and we are here to support government officials when they're ready to make decisions to continue to control this disease. Thank you.
Thank you very much, Claudia. We are now happy to take questions. Oh dear. Yes, please.
With a microphone? Or, yeah. Thank you very much, Michael Rabot, German Health Alliance. Thanks to all speakers. My question goes to Ab Osterhaus. Now, we've seen the spread, the enormous spread of the disease, but I was happy to see not a single spot in Namibia where we have one of the largest seal colonies at Cape Cross. How is that possible?
Well, we haven't seen that yet, and as a matter of fact, we are not sure if there's nothing there. Obviously, so it is. We have, let's say, Germany and the Netherlands. We have a good record in finding dead seals. Yeah, and if you find a massive outbreak in seals, then you will notice it, and it will be investigated. But we have a network. So if you have a couple of dead seals that die from an encephalitis, we tend to pick it up, yeah, because of the monitoring system. Obviously, when you have a virus that's spreading and causes mass mortality, like we have seen in Chile, for instance, then it will be picked up. So it's really a matter of, I wouldn't say it isn't there, but I would just say, well, perhaps we are not looking good enough, at least the single cases. And as you've seen in Northwestern Europe, the virus is not yet spreading from animal to animal, apparently, for which there are good indications in Chile at the moment.
Yeah, thank you very much. I'm Dr. Jean-Paul Uvoyo from Maltesa International. I'm very interesting for the presentation. Now I want to know for Dorothy, how many zoonotic disease in Ghana? And I think it could have been very good to make the whole assessment. The assessment for all disease, for all zoonotic disease, and to design the semi-contagion plan. It'd be very good for efficient resource, for optimize the resource, and for the population to involvement to the prevention of zoonotic disease.
Thank you very much for your question. In terms of the avian influenza, to the best of my knowledge until now, we haven't confirmed human case in Ghana now. However, there are other zoonotic diseases that we are doing surveillance, and some of them are AMR. We are doing tuberculosis, and my institute is working with which tuberculosis I myself am leading. We are working very well with the Veterinary Service Department and the National TB Control Program, looking at zoonotic tuberculosis. And AMR is one of the issues that we are also working with the same plus FAO. So there are some zoonotic diseases that we are doing. In terms of informing the population, there are some of the diseases we are working with the social scientists who are looking at how to engage the community properly in terms of our response, and that is ongoing. Thank you very much.
Hi, Nina Jamal from 4PAWS. We work on animal welfare. So we've heard about biosecurity and vaccination as measures to tackle highly pathogenic avian influenza. What about measures that are linked to the way that we farm animals? For example, fur farms. They're so animals are raised just for fashion. Or huge factory farms where you have thousands of animals, giving a chance for this virus to mutate. It's like a dream for a virus to be in a factory farm. What about taking measures to rethink our food system and move away from practices that are not necessary?
Maybe Up can take over this question.
Well, it's not one question. It was a series of questions, I think. Well, when we look back in history, we haven't seen these massive outbreaks of avian influenza, obviously. And the question is immediately, what has changed? And if your first question was about the fur animals. I couldn't agree more. We have seen with COVID, and we have seen, so really with flu, we see a similar thing there, and we allow the virus to adapt. So this is yet another reason to stop with these, at least this way of production. But that's my personal view, yeah, the first point. But I think from an infectious disease point of view, you're right. The second question, I'm looking also at my colleague here, Martin Beer. My personal opinion is that when we started to grow animals in the ways we are doing it today, without the proper biosecurity measure. I should say that there's an ethical question there that I do not want to address. But I think let's say the infectious disease question is indeed, how is it possible that whilst we have had these viruses all the time, why do we see this back and forth going of these viruses? There is one snag there, however, because I am sympathetic to what you said. However, when we look at free-range animals, for instance, so because that's an animal, that's an ethical question for animals there. Shouldn't we give them free? Shouldn't we have the animals in the free air and allow them to be free? And that is really, with the current pressure of the influenza, at least, that's a way to get the virus in, because that's not biosecure. So that's going against the ethical issue. So it's not that simple, and especially the question is, can we produce food? Can we produce food in the world? Can we produce meat the way we are doing it, or do we have to adapt it? And the obvious answer to the question is, if we cannot guarantee optimal or 100% biosafety and biosecurity, the answer is no, we cannot do it, because then the virus starts to spill back and forth between domestic and wild animals. So the answer is not so easy. I think there's different aspects there. We need sufficient meat worldwide. On the one hand, we have ethical issues, and I think we should come to a more balanced way of doing that. But I think definitely my colleague Martin Beer might have a slightly different opinion there.
As for the fur farms, I would see it exactly the same. We really have to have a close look there. This is mixing vessels we have not have before in this forum, so I would be very careful. I would be also careful to generalize it completely, because there's also backyard farming, which gives much more human-animal interfaces. So we really have to look step by step and say, okay, from animal welfare to biosecurity and risks. And H5 is a good example, because we had in Asia this ping pong system. And when the virus moved out to the ducks in the rice fields, then it was lost for intervention strategies. In the holdings, the H7 and 9 in China, it could be controlled by vaccination. So therefore, I would argue, yes, we have to have very close looks, but we also have to be careful not to generalize it, because otherwise we might lose trust. And therefore, have a close look and wise decisions.
I think this is quite a balanced view, and
as always.
And I tend to agree, Martin.
Thank you so much for the presentation. I really appreciate also you showing the data to back up the claims that were put forward. And so while obviously global health decisions are made in a reactive mode, what is going on at the moment, I think it's important to also look forward. And I've been hearing in your presentation also about transmission chains via physical contact. Just last month, it had been shown in a research article that actually my employer, I have to disclose this, Cell published that human isolated influenza was able to transmit between ferrets. And I was wondering how you advise global policy decisions to take into account, also to look forward, that we might have to have surveillance mechanisms that go beyond a physical touch. Thank you.
Would you like to? No, you won't.
Let Martin start.
Okay, I can start. I fully agree. So we need this genetic surveillance, which is important, but to learn also more about the viruses, we have to characterize them. Exactly how is transmission possible? And there is one, I would call it physical problem. We have too many reassortants. So we really have to concentrate, and we need new measures to be faster than in, for example, ferret trial. Because if you have 50 genotypes, you can select some, and the human ones are the first I would also select, or the dominant ones, but you might miss the most interesting one. So that's, I think many groups are working on this. Also organoids, for example. How could I guess the virulence, pathogenicity for humans, for example? At the end, if I want to look at the spread, I have to do the animal trial up to now.
Maybe I can briefly add to that, because I mean we know certain signatures, for example, in the receptor binding domain, which we can use for surveillance systems. But on the other hand, I think we should not only focus on the known culprits or on the known regions, because we know what influenza does best is adapt to new situations, to new receptors, which might come along with entirely different adaptive signatures. So I think a combination would be really important between the just hotspot screening in surveillance systems, but on top doing the functional assays, doing the ferret assays, because only in physiological situations we can learn whether the virus is really transmitting either by direct contact or by aerosols from animal to animal. So I think the combination is very important. Just having an in silico screen will not be sufficient. Up now, you would like to add, yeah?
Yeah, sorry. I denied it first. But basically, what we need to go for, of course, and there the ferret experiments are very important, as we have shown from the very beginning with the H5N1 studies we did in Rotterdam, that you go for correlates of transmission, for instance. Once you have those correlates, and as you said, you have to be careful, because new viruses might have different correlates, but the ferret experiments, although they are slow, I think they're so the transmission experiments, they are very important. And as a matter of fact, these were studies that were initiated already in the first half of the 20th century, already in England. Yeah, these studies were already done, and I think we cannot move away from them. The other thing is that the massive sequencing we are doing today, if you see the GIS8 and everything that's ongoing in human sequencing of human isolates, there's a lot of discussion at the moment, also for COVID, but also for flu. What is the optimum and where should we focus? Because there's a large focus in industrialized countries, in European countries, etc. But those areas where most of the things happen, there's relatively little sequencing. That was my last word.
Thank you. We have 1 more minute, and we can only have, I'm afraid, 1 more question.
Hi, my name is Theophilus Odum. I'm from Ghana, a GenWAC student, and I work with the Veterinary Service Directorate in Ghana. Thank you for the presentation. Listening to the presentation from Asia, it seems that the H5N1 is attaining different epicenter, and I think Africa is one of the destinations. Do you think that vaccination could be a solution to the new epicenters that is coming up? And from Ghana, what do you think could be the policy communication to the policy makers in terms of controlling avian influenza using vaccination as one of the solutions? Thank you.
Well, thank you for your question. You know, that empirical evidence to demonstrate that local epidemiological situation has drastically changed due to vaccination is quite limited. As Claudia presented, you know, that limited number of the poultry vaccination induced reduction in human cases is recorded. And otherwise, you know, in the last month, the EFSA, the European Food Safety Commission, produced the report about the recommendation for the possible response to 344B, and that used a mathematical modeling approach to combine different countermeasures. And what it has shown was that the intensive culling of poultry is not perfect in halting the transmission chains. And vaccination was what it appeared to be one of the best options to bring that ongoing outbreak under control. And in any case, as Claudia responded, there are a number of different types of the possible response to the coming outbreak, and combined interventions would be required. And relying vaccination alone is perhaps a dangerous policy as long as we do not understand the ecological dynamics, especially in wild birds. That's how I think.
So I'm afraid we have to close the session now. I would like to thank our experts for their excellent presentations, and I would like to thank the audience for the lively discussion. And yes, thank you. Have a nice day.